Welcome back to the Metabolomists. In this episode, I am joined by Rachel Kelly. Together, we discussed her work in the field of epidemiology, the place of metabolomics in cohort studies, and the work of COMETS to make metabolomic datasets accessible for epidemiological research. This episode also marks the beginning of our journey looking more closely at diseases through the prism of metabolomics, starting today with asthma, a term that includes a heterogeneous set of conditions with multiple presentations and phenotypes. We discuss how metabolomics can help us better understand asthma and hopefully soon provide better care for the patients who suffer from it. The Metabolomist is the podcast where we listen to the stories whispered by metabolomic data. I am Alice Limonciel, and this season we will examine the application of metabolomics in the clinics and the place of data interpretation in this field. Welcome to a new episode of The Metabolomist. Today I'm joined by Rachel Kelly. Hello.
Hello. Thank you for having me. Lovely to be here.
Thank you for being here. You're an assistant professor of medicine at Brigham and Women's Hospital in Boston and also affiliated to Harvard Medical School. Do you want to start by telling us about yourself, your expertise, and the research that you work on?
Yeah, thank you. I am at the Channing Division of Network Medicine, which is a joint appointment with Brigham and Women's and Harvard Medical School. The entire focus of our division is on omics technologies. My real interest is metabolomics. I first got into metabolomics through my PhD, and now I'm applying it mainly to metabolic epidemiology as a means to understand childhood health and development as my passion.
Okay. Did you use other techniques in your PhD? Like, do you see certain benefits to metabolomics, or what made you follow up with this method rather than others?
Yeah, so my training background is biology, and then I got into epidemiology, which I really found fascinating. And then I went to Imperial College in London to do my PhD in 2010. And, as I'm sure you know, Imperial's home to some of the leaders, some of the real greats in metabolomics. So I was lucky enough to go to many talks and courses led by some of these people. And it really just inspired me to get more involved in the field. And luckily for me, my PhD thesis was based around novel biomarkers of non-Hodgkin's lymphoma. And as part of that, I was part of the EnviroGenomarkers consortium. And EnviroGenomarkers was an EU-funded project that was aiming to find novel biomarkers of environmental exposures and relating those to disease. And one of the many facets that they had within EnviroGenomarkers was metabolomics, in addition to other omics and things. So I was able to learn from some of the top people at Imperial generating these data and incorporate that into my thesis. I think that the introduction via EnviroGenomarkers really helped me to conceptualize metabolomics, metabolites, and metabolome as this kind of intermediate phenotype between environment and disease. And coming from an epidemiology background, that was just very exciting.
I guess that was still a time where epidemiology had strong hopes for genomics to solve all the issues, right? And then metabolomics maybe came as a kind of savior for some diseases, at least. You experienced that firsthand in your career.
Yeah, I think so. I started my PhD and the title was molecular biomarkers, but we also were interested in genetic data. Like you said, as we've seen for so many phenotypes, so many conditions, I think the promise that we had, that we hoped for genetics wasn't quite living up to its potential in some cases,
There have been good examples, but there are still many diseases where we know that genetics cannot explain it all. And so I think it's really the advantage of metabolomics. You get a chance to see how the environment and the different factors can also influence it. So as an epidemiologist, I guess a really interesting method from that point of view. One of the topics we wanted to discuss is epidemiology and the use of metabolomics. Can you introduce the field of epidemiology just in a few words that we know clearly what we're talking about, that everyone is on the same page, and then we can see how metabolomics fits in that context?
There's multiple kind of definitions of epidemiology. And I think, epidemiology really had its moment in the sun with COVID, and suddenly everyone was an epidemiologist. In its broadest term, it's the study of the distribution and the causes or the determinants of health-related phenotypes in particular populations, and the application of this study to trying to control these phenotypes, these health problems. As epidemiologists, we're interested in the etiology of the disease, the pathogenesis of disease, and mapping the frequency pattern of health or disease states in specific.
And so from my point of view as a molecular biology, I also have this picture of epidemiology as a very statistics-heavy field. Is that right?
I think so. And, I think especially when you move to metabolomic epidemiology, which is, essentially just using metabolomics to address all those epidemiology challenges. And I think when you get to genetic epidemiology, it really is heavily statistics based, which is what I like about it.
Yeah, that's good. And in terms of challenges, since you mentioned it, when you started working with metabolomics, were there certain challenges either from the methodology itself or the data type or maybe the understanding of the results that really were striking for you at the beginning or maybe still now?
Yeah, I feel like everything, everything is still a challenge. You learn new things every day. It comes back to your question about statistics. We have these statistical approaches to deal with things, and a lot of those did come initially from the field of genetic epidemiology. When we started applying metabolomics, we were borrowing a lot of their techniques, a lot of their methods. Metabolomics is its own beast. It's very distinct, and you can't necessarily treat it in the same way that you would genetics. For example, thinking about something like the issue of multiple testing, which is a huge one for obviously genetic epi, but also metabolomic epidemiology. But again, you have to treat it slightly differently because you're talking about, depending on how your data is generated, the type of metabolomic data you have, tens, hundreds, thousands of statistical tests. But what we know is that they're not independent tests. And it's a great benefit that we know this, but we know that metabolites exist in these co-regulated and interconnected pathways, which means that the data is, it's highly correlated. And so you can't just do your kind of more standard on-for-any test. You want to try and take the structure of the data into account when you're dealing with it. So for myself and for everyone in the field, that's a challenge that we're all still working on. And people have suggested lots of great methods. I think there's a lot out there, but as a community, we still haven't really come to a consensus on the best way to deal with the data.
It's an interesting parallel you make because so for epidemiology as for metabolomics, it's great to have a strong statistical background, isn't it?
Yeah.
Then you're really well armed to work with it. Before we go to the paper we wanted to discuss together today, I wanted to ask you about COMETS because it's a good place here to discuss it, to explain what COMETS is and how it's organized, what it does, and also maybe how people can contribute their own data to it. So could you explain a bit for people who are not familiar what is COMETS?
Yeah, well, and firstly, we'd absolutely love to have anyone who has metabolic epidemiology data, we'd absolutely love to have you. So COMETS is the Consortium of Metabolomic Studies. It was actually initially founded as a national USA National Cancer Institute initiative. But they were just the funding body. From the outset, it really was designed to look at all different phenotypes. And the idea of it is to promote collaboration among people with metabolomic epidemiology studies. At the time that it was first founded, a lot of people kind of working in their silos. And I think one of the things with metabolomics is it's noisy data, but it can be expensive to generate. So, the bigger the sample sizes you can have, the better, the more in your results. So the idea was that to try and bring these people with these disparate studies together, especially to foster international collaborations and to provide a forum for discussion, development to address these issues, things like multiple testing, to kind of get everyone working together on these issues. And then the real thing that they really wanted to promote was this idea of doing meta-analyses, like bringing these studies together to just create bigger sample sizes. Same has been done in other consortiums, genetic epidemiology consortiums or whatever, but really just a way to bring these studies together. And it's still ongoing. From a few years ago, the membership is really exploding. I think at the last count, we had 81 cohorts from around the world taking part, which is, representing more than 150,000 people.
That's great.
Yeah, and from all over the world, the one, as is common to many omic studies, we're missing some representation from Africa, a lot of the Southern Hemisphere, really, but a good distribution of studies from Europe, from America, North America. So anyone who has a prospective metabolomic cohort, blood metabolomics and phenotype follow-up, it could be generated in either mass spec, it could be generated in NMR. We really encourage you to get in touch and join. Once you're a member, there's a huge number of benefits and opportunities.
And you mentioned, so it can be mass spec or NMR based, so you collect data. So I guess there are some limitations, but the platform is not in itself a limitation. But then you have to combine all this data together, right? And then there's also the Comet Analytics, I think, where users can analyze and filter through the data themselves. So you have to first combine all of that. And let's say if I measure my samples in one platform and then I measure other samples in another one, and I want to compare, let's say, a set of metabolites, first you have to make the connection between the different data sets that we can compare all of that. So that must be a huge amount of work, huh?
Yeah, yes, I'm sure everyone is very well aware. Combining metabolomics is a challenge beyond many other omic technologies. And, there are multiple reasons. We've got these different technologies that are measuring it. You've got even, if two being measured on the same platform, you're not always going to get the same coverage of the metabolome. Two different studies could get a very different set of metabolites. So that makes it difficult. Even simple things, and I think this is something. So we have two ongoing meta-analyses, one looking at age, one looking at BMI. And by far the biggest challenge has just been harmonizing the names across, because different companies, different labs, different scientists will call the, exact same metabolite something not huge. Well, sometimes it can be pretty different. Sometimes it's just the placing of the brackets or a comma versus a dash or whatever. And it just means that once you're doing it on a large scale, you can't just automatically merge them. You have to go through one by one and say, okay, it's the same metabolite as this. So there's that challenge. Oh, sorry.
There are people in the consortium then that are working specifically on doing that work, huh?
Yeah.
I guess. Yeah.
Yeah. When Comets was formed, and what they really wanted to do was make it possible for people to do these large scale meta-analyses. Like, well, how can we make that happen for people? Obviously, issues of data sharing, data transfer agreements and everything, it's complicated. They didn't want people to be, having to send data across and run it all themselves. So they wanted really kind of a centralized means by which these meta-analyses could be conducted. I really think it was genius. And the team, led by people like Steve Moore, Evie Matei, Ella Temprosa, Chris Zanetti, among many others, developed Comets Analytics to facilitate this. The way that Comets Analytics works is that the overall PI defines their research question. For example, we have a study, we want to identify metabolites that are associated with BMI. And so you go to the Comets consortium and you say to the cohort PI, do you want to be part of this meta-analysis? If they say yes, as part of Comets Analytics, they generated this kind of standardized input sheet. And so it's just an Excel file. It has different tabs, one where you put your metabolite information in, one where you put your subject information, and then one where you have these predefined models. So everyone's running the exact same models. The cohort PI populates the Excel file with their data. They run it through this web interface on Comets Analytics, which is extremely easy to use. I would recommend everyone to check it out. It's free for anyone can use it. You don't need to be a member. And then you get back your summary statistics, and those summary statistics also go to the cohort, to the overall PI of the leading the meta-analysis. Then the challenge is you've got these multiple different results files, but you've got potentially different metabolite names in all of them. So they do have a dedicated team, the data harmonization team, in addition to their many other work. They're automating it where they can. But for others, they're really going through metabolite by metabolite and saying, okay, is this the same as this? And they're using all of the metadata provided. So when the cohort PIs fill in that sheet, they're providing any names that they were provided by the lab, if they know mass to charge ratio, for mass spec data, if they know spectral information, if they have PubMed ID, Chem ID, whatever information they have, they include that in the sheet. So again, it's just a case of somebody going through manually and being like, okay, based on all this information, is metabolite X from this cohort the same as metabolite Y from cohort? So a huge undertaking that is constantly upgoing, because every time a new meta-analysis is run, every time a new cohort runs their data, they're then mapping those new data to the existing kind of master harmonization list. So an ongoing challenge.
Yeah, but then thanks to the work of the people behind this consortium, then you can really bring together the aspect of the community building, of people getting together and sharing their data, so that others can use it, but also they can benefit from the data of others, and also combining the data, which as a single lab, if you come from different platforms, is a huge undertaking. So I think it's really cool. Let's say if I'm thinking of doing some epidemiology work with Metabolomics, but I'd like to see what has been done or how promising it is for my disease, and there's some data in Comets already, I could also go in, give some minimum credentials, and then already mine what's already there in the database. Is that what you were saying with analytics, or you need to be a member to do it?
It doesn't work like that. It's more Comets Analytics is just an online web interface. When I say anyone can use it, what I mean is you can download the sample input file. You can populate the input file with your data, and then you can run Comets Analytics and get your summary statistics. But if you had a research question of interest, you can join Comets as kind of a full member if you have a cohort that fits the eligibility criteria. But you can also join as an affiliate member if you don't have data of your own. And then any member is welcome to submit what we call a project proposal. That proposal gets assessed by the steering committee just to make sure it doesn't overlap with anything that's already been done. If it's approved, which pretty much always is, then you can go to the cohort PIs and say, do you want to be involved in my research project? And that's when you kind of move to the step of using comets for the meta-analysis aspect.
Great, thank you. Is there anything else you'd like to mention about comets before we move on to the paper?
Just join.
Yeah, just join. And no, but it's true. If you have data and you'd like to share it with others and compare it with others, I think it's really a great way to do it.
Yeah, and the papers are just starting. You know, the initial paper describing the consortium came out a few years ago. The paper describing comets analytics is now out, and now we're really starting to get to the science papers. So those, more and more we're starting to get these meta-analyses papers out, and the results are very exciting. And it really does show that this consortium-wide approach, this metabolite harmonization approach that's kind of been developed by comets is really working well.
Yeah, that's great. Then that's the papers that I'm really looking forward to. You suggested one of your papers that I found really interesting. It's entitled Metaboendotypes of Asthma Reveal Differences in Lung Function, Discovery and Validation in 2 top med cohorts. And you're one of the 2 first authors of the article. And as the title says, it's about asthma. Can you tell us a bit what you were attempting to do in that paper? What were your main goals here?
The Channing Division of Network Medicine, where I'm based, we're really respiratory focused. We do a lot of studies in asthma and COPD. And so I had done many studies in asthma up until this point, omics and metabolomics of asthma. And I think one of the real challenges with asthma that we kept coming up against over and over again is that, like many common chronic complex conditions, it really isn't a single disease, and you can't treat it like that. And you can see that both from looking at the biology of it, but also even just, looking in the clinic, there is somewhat of a one size fits all treatment approach to asthma. And I think because of this underlying heterogeneity, it really hasn't been necessarily working that well for everybody. Not everyone is benefiting from these treatment approaches. And I think that largely comes down to the fact that, they all have slightly different types of disease. This was inspired by the idea of endotypes. It was something that was already looked at quite a lot in cancer in particular, but hadn't really been applied to a respiratory disease at this point. But we thought, well, rather than treating asthma as a single disease, why not try and group it into different subtypes? And
so those endotypes, right?
Yeah, people use this as an approach to kind of grouping on phenotype, but we were like, maybe it makes more sense rather to cluster people on their underlying mechanisms rather than on those phenotypic presentations, because then you're more likely to find biologically, mechanistically heterogeneous group, and then you can target treatment to that biology, to those mechanisms, rather than treat the symptoms of it, and hopefully make more targeted treatment for these different groups. Yeah, so that was kind of the idea.
This is what I found really interesting in this paper, because you use metabolomics, but you use it twice. So you use it the first time to create those kind of clusters, those metaboendotypes. And once you have them, then you look into what the differences are in terms of metabolites for these different groups to try and understand what's happening in each group that you couldn't see with the more usual clinical markers. So it was a really interesting approach, I thought. Maybe without going into too much detail, can you explain a little bit how you created those metaboendotypes? You made sort of networks, right, to create the different profiles?
Yes. So we used a method called similarity network fusion, which was initially developed for transcriptomic data. So this was the first time it had been used for metabolomic data, but it's a fantastic method that it just relies on underlying data structure. So it doesn't need to be specific to any particular data type. But as you said, the way it works is essentially it treats every individual as a node within this network. And then the edges that connect these nodes within the network are based on the similarity in metabolic profiles. So, the thicker your edge with another node, the more similar you are, metabolically speaking, to that node. So it constructs this network. And then we use spectral clustering to look for the most highly connected groups within that network. You end up with groups of people who are very similar to each other in terms of the metabolic profile and more different to individuals in these other clusters. When we did this, we ended up with 5 different clusters of individuals that we defined as metaboendotypes.
Yeah, I guess so. Then you have those groups, you've sorted your subjects in those different subgroups, and then you went and looked back at their phenotype to see if there were differences that you could find there.
Yeah, it was a completely unsupervised approach. We weren't conditioning on any kind of phenotype. So we just wanted to see, we didn't know that we would see differences. Our underlying hypothesis was people who were metabolically different are probably also phenotypically different. So when we looked at these groups, we found some of the important metrics for determining severity in people with asthma is lung function, essentially. So we found significant differences in terms of lung function between the individuals in these different groups. So in particular, we found a group who had the best lung function, who we define as kind of the least severe metaboendotype. And then there was a group of individuals who had the most severe asthma based on these lung function metrics. And again, the individuals in these two groups which show difference in severity were metabolically different. So that was, really exciting for us. Yeah.
Because you could expect, okay, so you have high function, low function, then you have high levels of those metabolites, low levels of the metabolites. But actually, these were different signatures that you found for those different groups. So then you have different stories happening potentially in each subgroup. I wanted to ask you a question about some detail about the method, because I noticed that you had 4 different metabolite platforms, so metabolomic platforms, and you treated them as kind of like 4 different omic data sets that you incorporated into a final data set at the end, or in a final network at the end. What was the benefit of doing that? Did you try first using everything together and then decided to split them because it was more efficient, or how did you get to this?
Yeah, that's a really good question, because this was something, we went back and forward on a lot. The idea is that you can build a network that is informed both by, say, gene expression and proteomics. So if 2 individuals that have a wide edge between them are more similar based on both those 2 data types. When we started this analysis, we only had the metabolite data available. So we're like, well, let's just see how it looks with the metabolites. And this metabolomic data was generated by Claria Klisch at the Broad Institute and by Robert Gersten, who's at Beth Israel. And this was generated as part of the TopMed initiative. So TopMed is transomic precision medicine for lung diseases. It's a national heart, lung, and blood initiative. So we had these data, and what was different about them was that the data generated by the Gersten lab at Beth Israel was a targeted platform. So we had 3 untargeted platforms, and then we had this one targeted AMI platform. So you're already dealing then with 2 quite different types of data. So we didn't want to just kind of put them together in a clump. We were like, well, we should probably be thinking about treating these as 2 distinct data types. So when we first did the network and we combined them all, we found that, the targeted platform was kind of dominating the construction of the network.
Okay.
At that point, it was hard to say, is it dominating it because it is biologically the most important platform, or is it dominating it because it's a targeted relative to an untargeted platform? So we decided that the best way to deal with that was to, go back to the basics of the similarity network fusion approach and treat them as though they were distinct omic data types, because the benefit of that is that you're weighting them all the same. I think there are arguments for and against. If you have 4 untargeted platforms, I don't think you necessarily would need to do it the way we did it. The other thing that I didn't say at the beginning, but I think is worth pointing out, is that as part of this TopMed initiative, we had data generated in 2 distinct cohorts at the same time. So we were able to...
I was going to bring that up too.
Okay. Yeah. So we were able to validate those findings. And again, I think that always gives you extra confidence in the method that you choose to use.
Yeah. You had 2 cohorts, one from Costa Rica and one from the US, both children. And you make those metaboendotypes, and it worked in the validation cohort. So you could reproduce the same groupings in the second cohort as well. And one other thing that caught my attention is that the metabolites that were used to form those networks and to define those endotypes, it was not, usually you see this kind of signatures is like 10, 15 metabolite stops, but you had something between 150 and 300 metabolites per group. And I wanted to ask you, was that a choice you made to take large signatures because you wanted that for some reason? Or did the model take you there somehow? Because I think it's also really powerful to have so many metabolites in the background. Maybe it helps to get a better resolution between the patients. Is that what happened?
Yeah, it's funny, as I was just on a call just before this, trying to work out probably a better way for determining the metabolic differences in the clusters. The way that it worked is we ended up with these 5 different clusters, metaboendotypes that we knew had different metabolic signatures. But that was based on, an unsupervised clustering approach. So we wanted to say, well, what really, what are those differences? What are the metabolites that are driving the differences that is making these individuals cluster differently, what are the key metabolites. And I think, there's a few different ways we could look at that. It was the best approach we had at the time. What we did was just a very simple logistic regression analysis. So we just did one endotype versus the other. So we looked at, our outcome was just endotype 1 versus all the other endotypes. We ran every model looking to see if a metabolite was significantly associated with membership of that metabo endotype. So that meant that a metabolite could be associated with membership of more than one endotype.
Sure.
I think it was interesting because it did give us that kind of long signature that we could then use in our enrichment analyses. We could try and understand the full pattern of it. But we're working with other kind of bioinformaticians to see if we can find a better way, because that answer is interesting, but I think there's probably other ways to answer that same question. So that's what we're working on now.
When you have more metabolites within one signature, doesn't it give you a kind of richness also to have so many metabolites that you get more precise about what this endotype means? I didn't see it as a weakness. I saw it more as a maybe why it worked out so well in that study, because it had so many metabolites. But you think it could have been less? Like now that you know maybe the data a little bit more, or new methods more, you expect you could do something similar with less metabolites in the end?
So I think, I should say that. So the formation of the endotypes, I think, should definitely be based on the whole metabolome. That second step we did, where we then did once we had metabotype membership as an outcome, we then did these regression analyses to try and identify metabolites associated with membership. So I think that second step we could probably refine to make more interesting. I think it comes down to that question that we come to a lot in metabolomics. Is this the purpose of this analysis to understand biology? In which case I would say you want as many significant, metabolites as possible. Or is our goal to identify biomarkers? In which case you would want a smaller number of unique metabolites. And I think that which one you do really depends on what your research question is.
But the question is what you're addressing here. So to discriminate between different subjects with different subtypes of a disease, and to have better targeted therapies, and to maybe find new therapeutic targets. I find the approach you had really relevant here. So much more than to have a collection of 1 or 2 biomarkers that would tell you, okay, you have asthma, but on 1 or 2 biomarkers, you will not be able to distinguish between 5 categories. It's unlikely, let's say, in the way that you did here. So I think for this, you explain it really well in the paper as well in the discussion. You're right, it has a twofold translational potential. And then for the reasons that we explained, so you can treat the patients in a much more efficient way, hopefully. So especially if we go to the second step where you try to understand these endotypes better, then you can hopefully find better therapies, or what is the best way to approach it first? Should we first attack the surfactant effect, or should we look at maybe more immune effects or things like this? And then, as we said, the new therapeutic approach, so the new targets that could be researched. And I think it works really well in this paper. And so the second figure that you have in the paper is chemical enrichment analysis. That's the approach that you took to take a deeper look at what's happening at the metabolite level. And there we see that a lot of those metabolites are lipids. So you have a lot of classes of lipids that have an important role here. And here again, if lipids are really so critical in the disease, then you're probably going to have to measure quite a few of them to have an idea of what's happening in the class. It's very different from the small molecule. So I think it's another reason why having a lot of metabolites in this case was probably a good approach.
Well, thank you.
So what was your experience with the chemical enrichment analysis? Why did you use this rather than maybe more classical pathway analysis, where it would tell you like this metabolic pathway is activated and so on?
Yeah, great question. And funnily enough, it was the suggestion of a reviewer who said, well, I think, we were initially using more classic pathway analysis. And I think you could have a whole podcast on the pros and cons of pathway analyses. But I think depending on the data set, I think that would be a great one. I would definitely listen. Yeah. As we know, there are cons to pathway analysis, and that really relates to things like the coverage of the metabolome. What are you actually measuring? What is your background reference data set? Because all of that has a huge impact on the pathways that you find, which can lead to results that genetically biased towards the better, the bigger, the more well characterized.
Exactly. And with the set that you see in this second figure, where you see there are lots of triglycerides and lots of different complex lipids, I guess you lost a lot of those metabolites in the pathway analysis, because it usually focuses on small molecules. So that must have been what rang the bell for the reviewer, I guess.
Yeah, exactly. Yeah. And so we were very grateful. I have to admit, it wasn't, I was aware of it, but I had never used it. And now we're, we're huge fans in our lab. We use Chemrit a lot. I do think it's a really nice method because as you say, it isn't reliant on these reference databases that are based on curated biological pathways. It's really based on the chemical similarity and the structure of the metabolites, which is hugely beneficial for lipids, which I think a lot of us are interested in, and which a lot of the data sets that are being generated contain. So I think it's a really nice method for that. And
and it's a great way to visualize it as well. You see immediately which are the more represented classes. On the other hand, if you're not an expert of biochemistry, you're going to have a lot of painful literature search when you go look into the small molecules. So I think both methods have their benefits, but if your signatures are like heavily reliant on lipids, it really makes sense to look at it that way. And then you went on to discuss, so I guess this was also one question I had. So you went on to discuss what's happening in each of the metaboendotypes, of course, and what that could mean in terms of mechanisms, in terms of what's happening for the patients at the molecular level. And so if you take the study from the beginning, so I don't know where it started for you, if it was like looking for cohorts, or if you also organized the collection of the samples. I'm not sure at what stage you got in, but like from the preparation of the study to the interpretation and the finalization of the paper, what would you say is the part that took the most time? This is a question I often ask. So
yeah, it's a great, and I'm always saying to the postdocs in our lab, because I think especially when they first start, they're always eager and itching to get papers out, and I'm like, science is slow, unfortunately. I should first of all say that these were two pre-existing cohorts, the camp cohort and the genetics of asthma in Costa Rica cohort. They've been long part of the data sets we have available at the Channing, and follow-up actually isn't even ongoing anymore on these cohorts, but we do have multiple samples. We'd already generated metabolomic data in both of them, but as part of the TopMed initiative that I mentioned, we were able to generate more. So, if you include data generation, it was probably 3 years start to finish. But I think from my point of view, because I'm not in the lab, I'm just the numbers person once we get the data. I think the thing that takes the longest for all these kind of machine learning studies and even more, classical statistics, it's really like calibrating those models. So for us, there was a huge amount of trial and error. We talked about looking at the data set as one versus looking at the 4 different platforms. And with machine learning and with statistical models, there really is no right answer. You're just trying to find the best fit for your data. So testing out different parameters, recalibrating the models, that was working out what makes the most sense, what fits the data best, what can we then validate in another cohort. That was probably the most time-consuming, but most important part of analysis.
Yeah, that's where the manual work really comes into play.
Yeah.
And considering the application that you make of metabolomics here, do you see a potential for it to be applied one day in the clinics, that people will use it? And if so, what does it take to get from the stage where we are now, so where we have this nice proof of principle, probably some independent researchers should show that they can also apply it to other cohorts or something like this. But let's assume this happened. What does it take to make it that one day we make a simple blood test in asthma patients and we can categorize them in one of those categories and make much better precision medicine for them?
I think there's a huge number of steps. I think the first thing, as you say, would just be making sure that we can validate them across time. This was based on a single blood sample. What we would want to do is to make sure that if we did the exact same thing in a blood sample from these individuals 5 years later, 10 years later, are they still clustering in the same groups? And if they're not, maybe that's because the phenotypes have changed. But I think, determining the stability of these, in addition to validating in other populations, is key to make sure that these really are biologically, clinically meaningful metaboendotypes. And then I think after that, the real work will be, as we talked about with the regression models, just digging down and being like, okay, well, what are the metabolic differences between these groups, and what part of that difference can we target? What is the real metabolic dysregulation in each of them, and what can we do to right that dysregulation? And then the next stage is clinical trials, moving into the clinic. I mean, developing new medications. I think, with all the work we do, I feel like we talk about clinical translatability because I do think potential is there, but I don't think for most metabolomic epidemiology studies, we're quite there. But you have to do the initial discovery work. You have to do the more basic science to inform those next steps.
Mm-hmm. And I think an approach like the one you took here is really interesting too, because asthma is far from being the only complex disease that has this heterogeneity. So a similar approach could be used for many different diseases, and it is being used, you said, in cancer, I think also for many other diseases. Is there anything else you wanted to discuss about this paper specifically that we haven't discussed yet?
Yeah, we're pretty excited about this approach overall, this idea that for multiple different, heterogeneous diseases, that you can try and identify mechanistically informative subgroups based on the metabolome. I really do believe in that principle. And I think we've shown it well in asthma. And now we're applying it to multiple other conditions that we have access to. One of the ones that we have collaborators at Mass Eye and Ear, and they're doing, we're working with them to do some really exciting work in looking at endotypes of age-related macular degeneration, a hugely devastating condition. We're seeing really great results there as well. I do think this is an approach that is exciting and that it's going to continue to show exciting results moving forward.
I hope so too. And you wanted to discuss another approach that you had in a second study on autism. Do you want to maybe quickly just say what was different there and what happened in that paper? We will put the link to the paper in the show notes so people can look at it too.
Yes, so this was a study where we got a grant from the Simons Foundation for Autism Research, and we plan to use the exact same approach. So they have a population of children, the Simons Simplex collection. So it's children with a diagnosis of autism spectrum disorder. And again, autism is a hugely heterogeneous condition that, has very varying phenotypic manifestations. And so we thought, why not take the same approach? Because there's a real strong rationale for why we might see metabolic different groups of people, groups of children with this condition, and why we might be able to, use the exact same approach to look at the metabolic differences between them. So we received the grant. We generated data on 2,600 children with autism spectrum disorder. But what was interesting about this was that we went in planning this approach, but the more we learned about the condition, we spoke with the people who generated the cohorts, we spoke with the clinicians, with the behavioral psychologists. We ended up completely flipping our approach. And instead, we clustered the children based on their phenotypic presentation, and then we looked for metabolic differences between the phenotypically different clusters. And I think it was interesting about that is it just goes to show that depending on the data that you have, depending on the population you're working in, the right approach differs for different data sets. Because again, we ended up seeing, we got 3 clusters of children with ranging phenotypic presentations of it was a very well, it's an autism cohort, so it was very well phenotyped. We were able to use a lot of data to inform the phenotypic clusters. And then again, we did see, significant differences in metabolites between these different clusters that kind of that tracked to those phenotypic differences, almost using the exact opposite approach. But again, seeing, that idea of clustering and looking at the metabolome, I think is just a really powerful approach to consider heterogeneous diseases, because at the end of the day, I think for a huge number of chronic or complex conditions, I think the real take-home message is you can't necessarily treat them as a single condition. You have to consider the idea that they are phenotypically different and therefore most likely biologically different, or there are biological differences.
And I guess with this approach also, you put yourself in a position where you have more chances to understand what is causing these phenotypic changes. So because you start from there, so you can more easily go back to that with the metabolomic results.
Yeah.
Okay, so we'll link to both papers on the show notes on the episode's webpage, so people can go and check them out. In this second paper as well, one thing I noticed is that the blood that was collected from the children was not fasted, which can be rather unusual for this type of study. And so for people who listen to the podcasts for a while, they might remember from last year that Hannah Laura Daniel mentioned that, as far as we know, there was no study that really determined that fasted blood samples were the best way to do anything. So, of course, they help to have a certain type of homogeneity for certain factors, or at least that's the hope. But here you have this set of non-fasted blood samples. And I wanted to ask you, what's your experience with this? Do you see a benefit to having fasted blood samples, or is it the same? Is it just a matter of how you handle it from the statistics point of view? What's your experience with this?
Yeah, the first thing to say is that we tend to deal with childhood cohorts or with pregnancy cohorts. So often our samples are not fasted just because they're populations that you can't necessarily ask to fast. So we often had samples that were either mixed in terms of fasting status or were not fasted. And I think, that's one of the things with metabolomic epidemiology is I think a lot of people are using historical cohorts, they're using biobanks, they're using data that wasn't necessarily collected for metabolomics. So often they're not going to be able to dictate the conditions in which the blood were collected. So a lot of people are going to have that mix or not necessarily know fasting status. And so I think it's a really important and interesting question to try and get a grasp on. How important is fasting status? Because it can affect the studies of many, many people.
Yeah.
What I will say from my own experiences, so as I mentioned, we have the BMI meta-analysis that we're doing in Comets. And so as part of that BMI meta-analysis, we have 45 different cohorts who are taking part, representing over 100,000 people. Many are historical, the blood was not initially collected with metabolomics in mind. So we have a mix of cohorts that were fasted and cohorts that were not fasted. What we have found, and this is specific to the question of BMI, but what we have found is that for the most part, it doesn't actually influence our results. So when we compare the summary statistics, the results of those cohorts that were fasted, for the vast majority of the metabolites, the correlation coefficients, the p-values are very, very similar. You're not getting a completely, between the fasted and the non-fasted, you're not getting a completely different set of metabolites. So we were somewhat surprised. We thought we would see more of an influence of fasting status, but actually we were very reassured to find out that we weren't, and that's not to say that some metabolites were not affected by fasting status. There was
exactly, because I guess there are some usual suspects that you would see differing, like glucose probably, or some triglycerides, or I don't know what you saw changing. Do you remember what were the differences in that case?
It was mainly lipids, to be honest. We would either be significant only in the fasted or only in the non-fasted. But I think that's kind of the beauty of metabolomics, and I think it's also something that we need to think about moving forward, is that similar to that issue of how do we treat multiple testing, I think we can't necessarily assume that we can treat every metabolite the same, because we know there are going to be some that are affected by things like fasting status, some that are affected by things like age. The issue of missingness is very different depending on what metabolites you're thinking about. So I think what we have to do as a community moving forward is work out, okay, what are those metabolites that are influenced by this factor or that factor, and consider treating them differently to the others. But, I guess the overall take-home message from that BMI study is, for the vast majority of metabolites, fasting or non-fasting in this analysis didn't really make any difference to our results.
Yeah, that's really a great example. Thanks. Okay, then I think we're ready to move on to learning about your favorite metabolite and why it is your favorite metabolite. I'm curious.
So this came from genetics initially. So we have another asthma cohort. It's actually a pregnancy cohort, VDAR, and it stands for the Vitamin D Antenatal Asthma Reduction Trial. So the idea was that pregnant women were randomized to either supplementation with vitamin D throughout their pregnancy or to a placebo, with the hypothesis that the mothers who were randomized to vitamin D were less likely to have offspring who had asthma. So that was how the study was initially set up. So we were looking at, for this particular analysis, we were interested in the children at age 3, once, they had been determined if they had asthma or not. And one, we were looking at from a genetic angle for this particular analysis. And one of the most replicated loci for childhood asthma is the 17q21, in particular the RMDL3 locus, the RMDL3 gene. And RMDL3 is a sphingolipid biosynthesis regulator. So essentially, we know that when RMDL3 is overexpressed, it kind of blocks the production of de novo sphingolipid synthesis. So we were interested in that gene, but then we got the metabolomic data available. So we started to look at it together. And what we found was really interesting was sphingosine 1-phosphate. Because, it plays such a key role in sphingolipid biosynthesis. So that is my favorite metabolite, because we came at it from a genetic angle, but it ended up being a real key metabolite in our analysis of these children with asthma, and in particular the vitamin D angle, because what we found was that in the mothers who were supplemented with vitamin D, their children overall did have a reduced risk of asthma. But it wasn't the case for everyone. There seemed to be this subset of children whose mothers received the vitamin D, but they still didn't have the reduced risk of asthma. And that all seemed to relate to the fact that those children had a specific genotype within the RMDL3 locus because it was overexpressed. They weren't producing enough sphingosine 1-phosphate, enough de novo sphingolipids anyway. So it didn't matter how much vitamin D you put into the system. Their levels of these protective sphingolipids were not increasing, and therefore their risk of asthma was not affected by the vitamin D.
Mm-hmm.
And so I think, when you think of sphingosine 1-phosphate overall, it's just like a really interesting metabolite. So that was kind of our first introduction to it. But now as we move more to looking at autism, mental health conditions, it's a really important metabolite in the body, and it's involved in multiple different conditions, and it plays multiple, different roles in immunity. And so I think there's a lot of interest overall in targeting this metabolite for multiple conditions. So that is why I chose it to be my favorite metabolite.
Great. Thank you very much. And it's also nice when you get a metabolite of interest that comes from different kinds of omics. I think that also gives you this idea that, this really is like a hub of what is happening in my study. And This is something, I don't know if you use multiple omics often in your work or if you really focus more on metabolomics, but it can also be a way to kind of give yourself a bit more confidence that this is really a change that is relevant in your study.
Yeah, as a division, we're really interested in multi-omic analyses, and it's something that we're trying to do more and more. There are just many analytical challenges involved. So I think, what we're really focused on right now is trying to solve some of those analytical challenges as we move to these more multi-omic studies. And it's funny because there's still so many challenges to solve just for metabolomics alone that you're magnifying when you start adding in multiple omics. I do think it's where the field is going. I do think it's the future. And I think it's huge, exciting potential. It's a learning curve. We're getting there slowly.
Yeah. Thank you very much. Thank you for taking part in the podcast and for your beautiful contribution.
Well, thank you. It was really wonderful. I really enjoyed it.
Thank you for joining us in this discussion. I hope that this episode gave you new insights and ideas on how to plan, conduct, and communicate your own metabolomic projects, and that you're excited for the future clinical applications of metabolomics. If you'd like to continue this journey with us, make sure to register for the Metabolomist email list on the podcast webpage, themetabolomist.com. If you want to learn more about how data interpretation is done, check out my book on the story principle at biocrates.com/thestoryprinciple. For regular news on metabolomics and data interpretation, you can follow me, Alice Limonciel, on LinkedIn, where I post on metabolites, analysis strategies, data processing tools, and more. And make sure to check out our other podcast episodes on the Metabolomist website.